So in this video, we're going to be thinking about the coronavirus and some of the new variants that you might have heard about in the news. And we're going to be thinking about that in terms of what that means of the biochemistry of what's going on with our viruses. So what we're fundamentally talking about at the moment is a process about cellular recognition. So if I have my little virus as my cartoon, and we know that the virus has got these spike proteins, these little blobs coming out of the side of the virus particle. So you've all seen the cartoons of that. So that's called the spike protein. What that spike protein is for is what it does is it binds to and attaches to receptors on your cell membrane. So this is your own cell, so it might be in your lungs, and there are receptors called the ACE receptors. And the spike protein binds to those receptors, and when it binds to the receptor, that starts it with the process of fusing with the membrane and then ultimately going inside the cell. So this recognition of the virus and the receptor is really, really important and as a potential target for action. So what I want to do is to think about that at a biochemical or molecular detail. So let's zoom in and think about this at a slightly different scale. So now I've got my virus. It's a much bigger looking particle. What we now know, because we've been able to actually solve the crystal structure, is that these little spikes have a bit more complicated structure. So what they have are 3 subunits. So they're what's called trimeric. They've got 3 subunits. And each subunit has 2 different bits of it. So it's kind of got quite a long extension from the cell surface, and then it's got a sort of more blobby domain at the end, and we call these the S1 and the S2 domains of the protein. So our virus has these. I'll just put another one to show you. All of those spike proteins in my little sketch actually look like this. So they've got 3 proteins coming together with these 2 different domains in them, and they, as I say, bind to the ACE receptor. So I'm just going to draw that in a bit bigger, that we find up the cell membrane. So that's the ACE. And that's the outside, and that's the inside of your cell, which is the cytoplasm. Okay. So in order for the virus to infect your cell and go inside the cell, what we really need to be thinking about are the interactions that are happening between these head groups, these S1 groups, and the ACE2 receptor. So there will be molecular interactions that are happening in that zone, and that's what we're thinking about. So this we call on the S1 protein is the receptor binding domain, also known as the RBD, you might see out and about. So we're thinking about the molecular interactions at this site, and the stickier that the virus is to the receptor, the more likely it is that it will stick to the cell and go inside the cell and infect you. So this is the absolute crucial molecular moment for understanding infection. So in order to do this, we need to understand a little bit about protein structure. So what is a protein and how are they put together? So proteins are built of amino acids. So these are small chemicals that are really important for you. So you get these from when you consume protein in your diet, for example. And they have the molecular structure of a central carbon atom, a carboxylic acid group, an amino group which contains nitrogen, a little hydrogen, and a group that is sometimes called the R group. And that R group can be basically whatever chemical group you like. You can stick anything on the side of an amino acid, but naturally there are 20 natural R groups. So each named amino acid has a different chemical group on its side, and that gives the amino acid different properties. The amino acids aren't free floating in protein. To make a protein, then they get bound together into these long chains. So what you end up happening is you've got N, C, C, N, C, C, N, C, C, and it goes on for hundreds of amino acids. You end up with these R groups sticking off to the side, and they usually alternate in which side they're going. So that's my protein chain. So we've got these different groups sitting off the side of it. So then if another protein chain comes along, which I'll just summarise in a sort of line diagram. So we've got an R group there, an R group there, an R group there. Then when you've got two different bits of protein coming together, then they can have an interaction via the R group. So if this is the virus and this is the ACE2 receptor, which R groups you have interacting really determine how attracted they are to each other and how sticky it is. So, for example, you could have two R groups one of which was an amino acid called lysine, which is given the code K. So lysine has an R group that ends in NH3+, so is positively charged. Now, if that's on the other side is next to an amino acid called glutamate, which is given the abbreviation E, well, glutamate's R group is a carboxylic acid, which would generally be negatively charged. So if you've got those two amino acids next to each other, positive things attract negative things, so you get a very strong interaction in there, and we call that an ionic bond. So that's an example of this R group and this R group. One is positively charged, one is negatively charged, and if they're in close proximity to each other, then they can interact and form a chemical bond, so they kind of stick together. So, what does this have to do with our coronavirus variants? Well, the UK variant has a mutation in it. So a mutation is where a bit of the genetic code is different, and that can give a difference in what's happening at the protein level. And there's a mutation called N501Y. Which is a mutation in that UK variant. So what this means is at position 501, so the 501st amino acid of the protein, there has been a change from an old amino acid. So in the old version, it was one particular amino acid with a particular R group, and it's been replaced with a new one. So the R group has changed on this amino acid, and it's at position 501, which happens to be smack bang at that receptor binding domain. So at the very surface of the S1 protein, that position 501 is right on the surface, right on the tip of the protein, and there's been a change in which amino acid is there. So let's have a think about that. So N is the amino acid asparagine. So in the old version of the spike protein, we had an amino acid that had this chemical structure. So it has a double bond oxygen and an NH2 group. And that's been changed for the amino acid Y, which is an amino acid called tyrosine, which has quite a different chemical structure. Actually, sorry, that OH should be there. So we've got different chemical properties on the protein. So the mutation gives us a different amino acid. So we've got a different R group. Which has different properties. And what that results in is different binding of the protein. So at this point, remember, we're talking about this point where the S1 protein is talking to the receptor. This version, the old version of the R group, there's 2 bonds to the ACE receptor. They're actually a type of, they're not this ionic bond, they're a type of hydrogen bond called a hydrogen bond, but they're still an interaction. So there's 2 bonds to the receptor with this version. With this version, the chemical analysis shows us that we have 5 bonds. So they've got a higher affinity for each other. Because of this different structure, we can make more bonds. That means that the bonds are stronger. So this interaction here, it's stickier. The S1 protein is more sticky. It sticks to the ACE receptor. If it's more sticky, there's more chance that that virus has got the time to bind to your cell, to get inside your cell and infect you. So this N501 mutation, it's not the only mutation in the UK variant, but it's quite an important one. Because we've changed one amino acid for another, we've gone from an asparagine to tyrosine, we've increased the amount of chemical bonding, so this new variant is stickier, it sticks to cells more, and that means that you're more likely to be infected, and that's why we're seeing increased infection rates with this new UK variant. So it comes down to the biochemistry of how the virus is attaching to your cell, and it's all about these R groups and how they interact with the receptor.